White blood cells or leukocytes are key participants in the acute inflammatory response, which is a multi-component process that defends the body against infectious organisms and ameliorates the impact of tissue infection. Now, among our white blood cells are monocytes, lymphocytes, neutrophils, basophils, and eosinophils. You can generally group white blood cells as granulocytes, monocytes, which can develop into macrophages or dendritic cells, and lymphocytes, with B lymphocytes capable of becoming plasma cells. By the way, granulocytes and monocytes are cellular components of what is called the innate arm of our immune system and are basically phagocytes, meaning they ingest invading pathogens such as bacteria. Lymphocytes, on the other hand, belong to the adaptive arm of our immune system. So basically, the adaptive arm is the one that's responsible for forming antibodies. Now, I will get a little bit into details on cells of the immune system's innate arm. Now, we have our monocytes, which are formed in the bone marrow and released into peripheral blood, where they differentiate into macrophages and myeloid dendritic cells after several days, normally in the order of 1 to 3 days. Then they migrate into tissues. Now, macrophages are monocytes that have migrated from peripheral blood into any tissue in the body, where they function by engulfing and killing pathogens. And upon doing so, they process these engulfed pathogens and present them as antigens in order to recruit other immune cells such as lymphocytes. Now, dendritic cells, on the other hand, are also antigen-presenting cells, but mainly to the T cells of the immune system, which is needed for activation of adaptive immune response. So you'll see in both cases, cytokines are produced for cell-to-cell communication. Now, collectively, monocytes, macrophages, and dendritic cells are members of the mononuclear phagocyte system. Now, macrophages have different names or aliases to them depending on the tissue wherein they are located. Now, many, if not all of these terms you've already encountered in first-year histology. Now, you can pause this video for a while to review on these different terms. Now, the other group of cells I would like to concentrate on for this topic is the granulocyte group, under which you have neutrophils, the most abundant of the granulocytes. Now, you learned from histology that neutrophils stain a neutral pink with H&E staining. Now, neutrophils are a type of phagocyte that are normally found in peripheral blood. Now, during the acute phase of inflammation, typically with bacterial infections, neutrophils are the first ones recruited or called to the site of inflammation, where they basically ingest and destroy pathogens within their intracellular vesicles. And as you probably already know, they are the predominant cells in pus, along with macrophages. Then we then have eosinophils, basophils, and mast cells. Now you'll notice their characteristic morphology on staining. They basically store granules that contain enzymes and toxic proteins that can be released upon their activation. So, eosinophils and basophils, along with neutrophils, make up your polymorphonuclear cell family or PMNs, which are basically mature white blood cells in peripheral blood. Now, let's step back a little bit and focus our attention back on phagocytes. Now, phagocytes have these so-called microbial sensors. And basically, when a pathogen enters, let's say, a break in the skin, for instance, it is then confronted by macrophages and other phagocytic cells possessing these so-called microbial sensors. And there are three major groups of these sensors. The first would be the TLRs, or the toll-like receptors. So these sensors on phagocytes recognize certain patterns on the pathogens. And we call these patterns PAMPs, or pathogen-associated molecular patterns. Now, basically, the macrophages have the TLRs and the pathogens have the PAMPs. And these PAMPs get recognized by the TLRs. And once this happens, there will be an initial cascade of rapid, robust inflammatory response. And this is due to cellular activation and cytokine release. Now, next you have NLRs, which stand for NOD-like receptors. So within that abbreviation is another abbreviation apparently. So the NOD here stands for nucleotide binding oligomerization domain. Now, NLRs are located in the cytoplasm and they serve as intracellular sensors for microbial products. And they activate this so-called NF-kappa B pathway. And hopefully you still remember from biochemistry, this is a signaling pathway that is very much associated with inflammation. Now, as a result, you have inflammatory responses similar to those initiated by TLRs. Now, sensors belonging to the third group are the RIG1-like helicases and MDA5 sensors. So again, more abbreviations here. The former stands for retinoic acid-inducible gene 1, and the latter stands for melanoma differentiation associated protein 5. Now, these are again intracellular sensors, but this time they recognize the viral single-strand RNA. The engagement of single-strand RNA with these sensors triggers the production of interferons, particularly interferon 1, and these interferons are highly effective inhibitors of viral replication. Okay, so we are still on phagocytes, but this time we'll be talking about phagocytosis, which is a multi-step process whereby a phagocytic cell, for instance a neutrophil, recognizes the pathogen, ingests it, and then destroys the engulfed organism. Now here we'll be particularly talking about the steps involved in this process. Now the first step involves phagocyte activation. Now all phagocytes, especially macrophages, exist in degrees of readiness. Now, macrophages are usually relatively dormant in the tissues. However, during an infection, they receive signals in the form of inflammatory mediators. So you have a bunch of them listed here. Now, these inflammatory signals then trigger an increase in their production of major histocompatibility complexes, or MHC type 2 molecules. And this prepares them for presenting antigens once they ingest the pathogens and process them for antigen presentation. Now, once a pathogen enters the blood or tissue, the phagocytic cell migrates to that site following activation. They call this phagocyte movement or migration diapedesis. And this migration is dependent on the release of chemoattractant signals produced by either the cells of the host or the pathogen itself. Now, interleukin 8 and 17 are particularly effective chemoattractants, especially for neutrophils. Now, fibrin split products, kinins, and phospholipids are also effective for this purpose. Now, in the initial stage of diapedesis, neutrophils attach to the endothelial cell surface by means of adhesion molecules such as P-selectin. Now, neutrophils then follow the chemokine attraction and migrate from the circulation through the endothelium into the tissues and to the site of the infection. Now, one thing to understand about phagocyte migration or diapedesis is that cell surface proteins, particularly on the vascular endothelium called integrins, facilitate this process. So basically, the adhesion of leukocytes or phagocytes to vascular endothelial cells is mediated specifically by transmembrane glycoproteins of the integrin family, as well as the selectin family too. Now, the next step in the phagocytic process involves attachment of the phagocyte to the pathogen. Now, the attachment can be unenhanced, and this involves interaction between pattern recognition receptors and PAMPs. And by pattern recognition receptors, I'm referring to the TLRs or toll-like receptors that I talked about earlier. Now, the phagocyte to pathogen attachment can also be enhanced, which makes the phagocytic process more efficient. Now, this enhancement occurs when antibodies are available to coat the surface of bacteria and facilitate their ingestion. Now, this process is called opsonization. Now, opsonization can occur via antibodies alone, acting as opsonins. Now, IgG is an example of such antibodies. Now, there will also be antibody-antigen interactions that trigger the complement system via the classic pathway to generate opsonin. I'll be talking about the complement system later in this video. Now, opsonin can also be produced when the alternative pathway is activated and C3 is generated. Now, the final steps involved in the process of phagocytosis are the ingestion and destruction of the pathogen. Now, acidification, which occurs within the phagosome, is used to kill the ingested pathogen. Now, the phagosome pH is 3.5 to 4, and this level of acidity is bacteriostatic or bactericidal. Now, once the whole process of phagocytosis is finished, the neutrophil then undergoes apoptosis and dies. Now, basically, this figure summarizes the whole process of phagocytosis. It depicts the destruction of an ingested microorganism, which is represented here in this figure in orange. Now, starting with letter A in this figure, the neutrophil binds an antigen molecule on the opsonized microbe via a receptor. Next, you see the neutrophil enveloping the microbe. Afterwards, secretory granules, which are the green circles in this figure, fuse with the newly internalized phagosome, delivering their proteolytic contents. Now, the enzymes and cytotoxins contained in these granules then destroy the microorganism. Now, in the final figure, any remaining debris from the destruction of the invading microbe is then expelled. It doesn't stop there though. Leukocytes, as well as B cells, absorb these expelled fragments via endocytosis, and then afterwards direct them to the cell surface in association with a membrane protein called the Major Histocompatibility Complex, or MHC for short. So basically, these fragments are now what we consider as antigens, which are presented by these leukocytes, which are now called antigen-presenting cells, to lymphocytes via the MHC, or the Major Histocompatibility Complex, on their surfaces. Okay, so I guess at this point we should define what an antigen is formally. So an antigen is any substance that may be specifically bound to an antibody. So these antigens could include simple intermediate metabolites. They could include sugars, lipids, hormones. Basically, they're macromolecules, and this could include complex carbohydrates, phospholipids, nucleic acids, and proteins, perhaps from the invading pathogen. Now, basically, only macromolecules are capable of stimulating B lymphocytes to initiate hormonal response. Now, there can be some substances, some small chemicals, an example of which would be dinitrophenol, which may bind to antibody, but they cannot activate B cells on their own, meaning they're not immunogenic. So that means they are called haptens. And when it comes to haptens, haptens need to be attached first to a macromolecule or a carrier macromolecule in order to initiate an immune response. Okay. Now, the hapten carrier complex can act as an immunogen. Okay. So you have to have both the hapten and the carrier, the macromolecule carrier, in order to mount an immune response in this case. Okay, so besides formally defining what antigens are, let's also formally define major histocompatibility complexes, or MHCs. So when we talk about MHC, this actually refers to a group of genes involved in the immunological recognition of self and non-self. So these are found basically in all or virtually all vertebrates, and the MHC is actually encoded in or located in chromosome 6 in humans. Now, the MHC molecules or proteins that are produced by these genes, they bind peptide antigens and present them to T cells via the T cell receptors. And this is actually responsible for T cell antigen recognition. Now, human MHCs can also be called human leukocyte antigens, or HLA complexes. Okay. Now, MHC exhibits a high degree of polymorphism. Okay. It means that different individuals generally express different MHC alleles or allelic variants. Okay. Now, allow me to talk a little bit about T cell receptors, because there's actually something interesting about TCRs. So, TCRs only recognize peptide antigens presented in the context of the MHC molecule on the APC. And what is very interesting about the structure of the T cell receptor is that they have this structure similar to that of antibodies. Okay, so it has a constant and a variable domain. Okay, so basically this arrangement is similar to that of the chains of antibody molecules. Okay, so this is a receptor that actually has structures of antibodies. Okay, and T cell receptors are actually quite specific for antigens, but the antigen must be presented on a self-MHC molecule to which the TCR is also specific for. And you call this very specific setup as MHC restriction. Okay. Now, should this antigen be presented by another allelic form of the MHC molecule, then the TCR does not recognize the complex. Okay, so that's how specific it is. So as I mentioned in passing earlier, so there are actually two important classes of MHCs. So you actually have MHC class 1. So this is actually expressed in nearly all nucleated cells in the body, maybe except for the retina and the brain. And there's also MHC class 2, which is encoded by the HLA-D region in our genome. So under this group, you have three main families of molecules that are encoded. So you have the HLA-DP, HLA-DQ, and HLA-DR. Now you encounter these groups again later in pathology because there are certain diseases that are implicated by these particular groups of molecules. Now, MHC class 2 complex is actually located on APCs, right? On macrophages, dendritic cells, B cells, any antigen presenting cell. Okay. Now, there's actually a third class of MHC, and a gene locus. This is actually a gene locus that encodes for complement proteins and several cytokines, okay, which are all involved in the immune response. Okay, so now again, still with major histocompatibility complexes, it's best that I present this particular image here. So the panel on the left is what I've been talking about earlier. This is the usual setup for antigen presenting cells, wherein the antigen, which is processed from the engulfed pathogen, is presented in this so-called peptide binding groove in the MHC. Okay. So now you can also have certain types of antigens, and we call this super antigens. So these are actually larger molecules, and what they do is they actually bind the class 2 MHCs outside of the peptide binding groove, as you can see here on the image on the right panel. And basically, it also interacts with the T cell receptor, right? Okay, so and then this apparently can also elicit an immune response. Now, as you probably have figured out already from the images that I showed in the previous slide, so super antigens do not require processing, like the usual processing that happens when you engulf a pathogen and then you derive the antigen from it and you present it on the MHC. Now, with super antigens, they are able to bind the MHC molecules outside of the peptide binding cleft or the peptide binding groove, as you have seen in the image in the previous slide. Now, some bacterial and viral antigens are capable of activating a large number of T cells through this particular pathway. So here are some examples. So staphylococcal enterotoxin can do that. The TSS toxin can also do that. You can also have group A streptococcal pyrogenic endotoxin A. And this is actually the pathway that is implicated in the so-called cytokine storm, wherein you have actually a huge amount of cytokines being elaborated in the immune response, and this actually is responsible for septic shock and multi-organ failure, and we've actually described this quite a lot in COVID-19. Okay, so we are done discussing the granulocyte group and the monocytes, all of which are phagocytes and subserve the innate arm of our immune system, as I mentioned earlier. We are likewise done discussing phagocytosis and the basics of antigen presentation by antigen-presenting cells via major histocompatibility complexes or MHCs. Let us now look at an overview of the other arm of our immune system, which is called the adaptive arm. Basically, the adaptive arm has two responses, the antibody-mediated response and the cell-mediated response. So, in the antibody-mediated response, CD4 T lymphocytes recognize the pathogen's antigens bound to the class 2 MHC molecules on the surface of an antigen-presenting cell, which can be a macrophage or a B lymphocyte for that matter. Now, as a consequence of this interaction, cytokines are produced that stimulate B lymphocytes to express antibodies that display specificity for the antigen. Here, the B lymphocytes undergo clonal proliferation and differentiate into plasma cells, which in turn produce antibodies. Now, in the cell-mediated immune response, on the other hand, the antigen presented on the surface of the antigen-presenting cell by way of MHC class 2 complex is recognized by the CD4 T lymphocyte, whereas the antigen MHC class 1 complex is recognized by CD8 T lymphocytes. Now, both subsets of T cells produce cytokines, become activated, and expand by clonal proliferation. Okay? The CD4 T cells that develop stimulate B cells to produce antibodies and promote delayed hypersensitivity, while the CD8 T cells direct their activity mainly at the destruction of cells in tissue grafts, tumor cells, or virus-infected cells. Okay, so I guess I should segue the discussion now onto B lymphocytes, which, as you know, are involved in humoral immunity, and this is actually the antibody-mediated arm in the adaptive immune response. Now, each individual has a large pool of unique B cells, and this is usually in the order of 10 to the 11 or 10 raised to the 11th power. And here are locations wherein you can find B lymphocytes. So you can actually review them, and these actually, I believe, have been already covered to some degree in basic histology. Okay. Okay, so this slide here explains the whole concept of clonal selection. Now, of course, this is not too detailed because obviously, once antigen binds to the B cell receptor, of course, there has to be some antigen presentation to T lymphocytes in order for cytokines to be released and proliferation of B cells to happen. Okay, so this is really just a simplified image just to show you the whole concept of clonal selection. So I actually have here a set of B cells. So you have different types or different forms of antibodies on different B cells. And this is actually represented by different colorings of the B cell receptor on each B cell. Okay, so basically bottom line is you have many B cells and each B cell actually has a different form or different shape of their surface B cell receptor. Now, an antigen from the pathogen, from an invading pathogen is present. Okay, so you have an invading pathogen and you have that antigen on that pathogen. So because you have all these different sets of B cells, the antigen is bound to bind to a particular B cell receptor on one particular B cell. And once that happens, of course, that particular cell would now be committed to proliferate. So this is actually the whole concept behind clonal selection. And because of this high specificity, you have to have many lymphocytes that have very different shapes of their B cell receptor in order to accommodate a whole wide range of shapes and sizes of antigen. Now, once that antigen binds to a specific B cell, so that B cell now undergoes clonal selection, it proliferates. Okay, so as you can see in the subsequent images, there's proliferation of that particular B cell, and many of these B cells differentiate into plasma cells to produce the antibodies against the pathogen. But as you can see, there's actually a subset of these B cells that differentiate into memory B cells because the importance of this is once the initial infection has been resolved, if a secondary infection happens with the same antigen, then you already have these particular memory B cells that can actually be able to skip this whole proliferation process, this whole process initially discussed under primary response. So during a subsequent infection with the same pathogen with the same antigen, okay, so the memory B cells would now be able to help skip a few steps in this whole immune humoral response. Okay, so you now have directly differentiation. You now directly have a proliferation of these memory B cells and direct differentiation to plasma cells to produce the antibodies. So now you have a quicker response second time around. Okay. Okay, so that's it with B lymphocytes and the different classes of and functions of your antibodies. So I guess at this point we can now move the discussion on to T lymphocytes. So I guess in order to understand the functioning of T lymphocytes more, we probably should review T cell development. So as we all know already, so T cells are derived or arise from T cell precursors. And basically, this happens in the bone marrow, and afterwards, they move on to the thymus, particularly first in the cortex, where there's now beginning differentiation between your CD8 positive and CD4 positive T cells. Okay, Now, in the thymus, particularly in the cortex of the thymus, that's where you initially have the site for positive selection, meaning those cells that are actually able to recognize self-peptide plus self-MHC, at least at a very weak affinity, are allowed to survive. Okay, because as you could recall from physiology, it's actually in the thymus where you actually quote unquote train your T cell to be able to recognize between self and non self. Now, initially, you gather all of the T cells first that are able to at least weakly bind to your self peptide and your self MHC. Then, afterwards, once you move further into the medulla, that's where your site of negative selection happens. Now, only those T cells that manage to form a strong or high affinity between your self peptide and your self MHC are allowed to survive. Those with weak affinities are killed off basically, right? And now, so basically, you've pretty much ensured that your T cells are able to recognize uh, your own tissues yourself. Okay, and then from there on, so CD4 eventually becomes your helper T cell, and depending on the cytokine stimulation, you can actually differentiate your helper T cell into Th1, Th2, Th17, and Th subreg. And your CD8 plus cells or your CD8 positive cells eventually become your cytotoxic T cells. Okay, and all of this differentiation processes beyond the thymus occurs in your lymph node. Okay. Okay, so now recall that your mature T cells will be really your CD4 and CD8 cells, or T cells rather. Okay, now let me now segue into the topic on cell proliferation and differentiation of these mature T cells. So let's first look at proliferation and differentiation of CD4 T cells. So basically, this involves something called T cell co-stimulation. Okay, so you initially have a naive T cell, right? And you have an antigen presenting cell that presents the antigen to the naive T cell via its MHC class 2 complex or MHC class 2. Okay, now basically, just to describe a little bit what this co-stimulation is. So T cells require two signals to become fully activated. Okay. Now in this case, the first signal here is what you see on the left side of the image. So basically, the first signal is what's called antigen specific, and it is provided through the T cell receptor on the naive T cell, wherein that T cell receptor of the naive T cell interacts with the MHC class 2 of the antigen presenting cell. Okay. Now, there actually is a second signal that happens. Okay, so this second signal, which is the co-stimulatory signal, is antigen non-specific and is provided by the interaction between co-stimulatory molecules expressed on the membrane of your APC and the T cells. That is why you see here on the right side of the image, you already now have two interacting proteins between the two cells. Okay, so there's now co-stimulation. Okay. One very well-known type of co-stimulation would be, or rather one of the best characterized co-stimulatory molecules expressed on T cells would be the CD28, okay, which interacts with your CD80 and CD86 on the now activated T cell. Okay, so basically the interaction between your CD28 and the CD80 and CD86 between the APC and the activated T cell actually facilitates T cell activation, proliferation, survival, and cytokine production, as mentioned here in the image in your screens. So basically, there is now secretion of your interleukin 2 and interferon gamma, and there's also increased expression of your interleukin 2 receptors. Okay. Now, basically, what I just described are those for CD4 T cells. Okay, but how about for proliferation and differentiation of CD8 T cells? Now, if with CD4 co-stimulatory signaling is required, apparently this co-stimulatory signaling is not really required for effector CD8 T cells. Okay, so there's really no co-stimulation that is required for this. Now, I'll spend the last remaining slides discussing the different T cell effector cells and their functions. Now, I mentioned earlier that CD4 cells, or particularly your CD4 effector cells, given the correct cytokine stimulation, can differentiate into Th1, Th2, Th17, or Thregs. Okay, so I'll be going through that in further detail here. So CD effector cells, so you have emergence of your Th1 type, and that follows appropriate triggering by your interleukin 2 and interleukin 12. Now, they either activate macrophages or cause B cells to switch to produce different subclasses of IgG. And by that we mean cause B cells to switch into plasma cells, because really that's the only way that beta cells will be able to produce immunoglobulin. That is by having them differentiate into plasma cells. And your Th2, on the other hand, its formation is stimulated by interleukin 4. And as far as function goes, they activate mast cells and eosinophils. Okay, and bottom line is they cause B cells to synthesize IgE. So you can now see here that this is actually related to anything that involves anaphylaxis, okay, or anaphylactic reactions for that matter. Okay, so now moving on to the other CD4 effector cell types. So the emergence of Th17 type of effector cells follows activation by your transforming growth factor beta, your interleukin 6 and your interleukin 23. Now, what happens here is your function-wise, your Th17 is the one responsible for producing a variety of interleukins, particularly your interleukin 21, 22, and 17. Now, these particular interleukins, they stimulate stromal and epithelial cells to produce IL-8, as we learned in the previous video, is a very strong chemokine, right? And clearly, as you can see here, when would you actually stimulate stromal and epithelial cells to produce such? Only when they are infected by an invading pathogen, okay? Because of course, there has to be a way in which the infected epithelial and stromal cells are able to call on the cavalry to effect defense against these invading pathogens. Okay, and then we have your Tregs. So as the name implies, Tregs are actually involved in probably suppressing the immune response, because you don't want things to go overboard as well. Okay, because remember, the immune response is a highly inflammatory process. And of course, it can be double-edged, meaning it can also harm the host. So there has to be a way, at least in healthy individuals, that you are able to regulate the functioning so it doesn't really go into hyperfunction. Okay, so you call on your Tregs for that. Okay, so the Tregs basically develop when they're exposed to transforming growth factor beta alone, not with the other interleukins that you see in the previous type. Okay, and then so these particular Tregs, they express CD4 and CD25 as well as a cell surface marker called FOXP3, right? And they basically produce, of course, more TGF beta and interleukin 10, and apparently these two in concert are responsible for suppression of your immune response. Hence, the counterbalancing of the pro-inflammatory effect of the whole immune response itself. Okay, so hopefully that was clear. Okay, so this should be the last slide for this particular video lecture. So I just discussed CD4 effector cells. So there are 4 types of those depending on the appropriate cytokine stimulation to affect that type of differentiation. Now let's now go to CD8 effector cells, which I have shown in a previous image. They're the ones that eventually differentiate into cytotoxic cells, right? And as you could recall, so basically it involves the interaction between your T cell receptor and class 1, okay, class 1, not class 2, class 1 MHC, okay? So basically the class 1 MHC antigen complex, which is present on the antigen presenting cell, when it interacts with your TCR on your CD8, they eventually differentiate into cytotoxic cells. And basically, it the usually it are these the cells that are actually presenting the antigen via class 1 MHCs typically are those cells that are infected with the pathogen. So this particular interaction causes the CD8 effector cells to be cytotoxic in nature, and it would actually kill that particular infected cell, right? And these types of cells, your CD8, and I mentioned already some of them in the previous slide. So they contain granules that contain perforin, granzymes, and granulosin or granulosin, or not really sure what the proper pronunciation for that would be. But bottom line, these powerful enzymes are responsible for perforating holes in the cell wall of the pathogen and for ultimately destroying the cell that is infected by these pathogens. Okay? Now, let's backtrack again to our phagocytes. They actually have a special ability in that they're able to effect what's called a respiratory burst. Now, during this so called respiratory burst, these phagocytic leukocytes rapidly consume molecular oxygen in order to produce reactive oxygen species, which are major components of their chemical and enzymatic arsenal used to destroy ingested pathogens. Now, the first step in the formation of microbicidal reactive oxygen species during the respiratory burst is the synthesis of superoxide, which is catalyzed by the NADPH oxidase system. On the other hand, there's another enzyme system called myeloperoxidase, which catalyzes the formation of oxidants that contain chlorine, such as hypochlorous acid or HOCl. Now, myeloperoxidases contain two iron heme-like centers, which are responsible for why pus or phlegm gets its greenish color. Now, this figure, which was taken from Mark's Basic Medical Biochemistry, summarizes the whole process of the respiratory burst. Now, activation of NADPH oxidase initiates the respiratory burst with the generation of superoxide. Superoxide then gets converted to hydrogen peroxide by superoxide dismutase, if you could still remember from the first video of this series. Now, here you also have granules containing myeloperoxidase getting secreted into the phagosome containing the invading pathogen. So, myeloperoxidase is the one responsible for producing hypochlorous acid. Now, hydrogen peroxide can also go on to generate other reactive oxygen species such as hydroxyl radical. At the same time, you also have nitric oxide getting converted into reactive nitrogen oxygen species, which, along with hydroxyl radicals, destroy the invading pathogen. Now, the entire process of the respiratory burst lasts only 30 to 60 minutes. Now, when you have mutations that result in impaired function or absence of NADPH oxidase, chronic granulomatous disease develops. So what happens here is that there is impairment in the production of reactive oxygen species. And because of this, phagocytic leukocytes are now unable to mount a respiratory burst to kill ingested pathogens. Now, chronic granulomatous disease is a relatively uncommon clinical entity, but individuals who have it commonly present with recurrent infections. They also form granulomas, which are chronic inflammatory lesions in the skin, lungs, and lymph nodes, as a means to wall off invading pathogens. I'll show you a histological image of this in the next slide. Now, here is what a granuloma looks like under the microscope. So basically, in an attempt to wall off the infection in chronic granulomatous disease, epithelioid cells or macrophages fuse together to form what's called Langhans giant cells, which are considered pathognomonic for granulomatous conditions. Now, granulomas are likewise characterized by central caseous necrosis. So, a more common condition called pulmonary tuberculosis also presents with granulomas, but often the area where the central caseous necrosis is would contain acid-fast tubercle bacilli. Now, you learn more about this in your pathology subject in the second year. Now, this ends the video series on RBCs and WBCs. Congratulations for making it this far. Now, just as a reminder, please read the two corresponding chapters in Harper's, as well as the article for required reading in preparation for your modular exam. Thank you very much.
pharmacists. Thanks for working the front line. Thank you, scientists. Thank you, pharmacists. Thanks for working.